Good morning, everyone. Today is the fourth day of the 14th HOPE meeting. First, we have Dr. Johann DEISENHOFER's lecture. Let me introduce the moderator for this session, Dr. Michi Suga from Okayama University. Please welcome Dr. Suga.
Good morning. I'm Michi Suga from Okayama University, the moderator for Dr. DEISENHOFER. It is great pleasure to introduce him to the HOPE meeting. Dr. DEISENHOFER is currently Professor Emeritus at UT Southwestern Medical Center. He received a doctoral degree in experimental physics in 1974 from the Technical University Munich. And then he worked at the Max Planck Institute, where he succeeded in the structural analysis of integral membrane protein involved in light harvesting and energy conversion in photosynthesis bacteria. He received the 1988 Nobel Prize in chemistry for the structural analysis with Helmut Michel and Robert Hoover. In 1988, he moved to Dallas to have his lab and have made many contributions to the structural biology field until now. Here, I emphasize 2 points about why his research was significant. The first is the protein he analyzed is called a photosynthetic reaction center, which contains a special pair of chlorophylls that can convert light energy into chemical energy. Obviously, photosynthesis is an essential biochemical reaction for all living things on Earth. Second, the photosynthetic reaction center was the first structure of integral membrane protein to be analyzed at atomic resolution. As membrane protein was embedded in biological membrane, and it must be extracted from the membrane using detergent to analyze the structure. At that time, many researchers believed that structural analysis of membrane protein was challenging and almost impossible. Therefore, his pioneering work led to many researchers participating in the X-ray crystallography of membrane proteins, and various excellent studies have been achieved afterwards. Dr. DEISENHOFER, please start your presentation when you are ready.
Thank you very much for these friendly words. And I want to thank the organizers for inviting us, and I thank all of you for listening to what I want to say. Here you see the institution I come from. It is an old picture. It is one of the most fastest growing university medical schools I know about. And when I came there, it had about 5,000 employees, and now it has close to 20,000. Anyway, I want to, as Michi mentioned, I want to talk about something that came before I moved to this place. And it is about photosynthesis, and in Monday morning we already heard that that photosynthesis is a type of catalysis. So we have a hot body, the sun, 5,800 Kelvin approximately, and we have a cool body, the Earth, 288 Kelvin. And we have in the atmosphere or on the surface of the Earth, we have carbon dioxide and water, and the light energy converts this into oxygen and sugar or carbohydrate. And there's the process by which this happens is called photosynthesis, obviously. And it can be reverted, among other things, by cell respiration, consuming oxygen and carbohydrate and producing CO2 and H2O. And this, of course, is these rectangles represent the catalysis. In the case of photosynthesis, first, I would like to show this to emphasize that the Earth atmosphere was not always as it is today. This is a diagram from a review that appeared a while ago. And it represents the concentration of oxygen in the atmosphere. And if you just follow what they call standard model and start here approximately where the authors believe the life appeared on Earth, then for a long time, the oxygen concentration was very low, in the order of 10 to the minus 5, 10 to the minus 6 of the present concentration. And then happened an event that is now called the Great Oxygenation Event by the geologists about 2.3 billion years ago, where the oxygen concentrations increased by, I think, 4 or 5 orders of magnitude to reach about 1% of what it is today. And the blue curve indicates possible modifications and refinements, but in principle, this is what happens overall in a long-term average. And there was about 500 million years ago, there was another such event where the oxygen concentration rose again by about a factor of 10 to 20. And only then, only in this situation, in this state, did it make sense for organisms to breathe, because before that, the oxygen concentration was far too low. However, one has to keep in mind that life up to about here all happened in the ocean. There was no significant amount of life on the surface, on land. And I guess the opinion is that at this point in time, the oxygen concentration could increase because plants first evolved and then moved to the surface and on land, and then began their existence in a much higher intensity of sunlight. But it is clear that there is no other known process apart from photosynthesis that could have caused these increases. And therefore, photosynthesis, and not only therefore, but also therefore, it received a lot of attention for a long time as soon as the technical and experimental possibilities existed, people tried to figure out how it works. And they came up, first of all, finding out where in plants, for example, photosynthesis is happening. And this is in so-called chloroplasts. This is, I think, from a textbook showing a part of a spinach leaf. And this chloroplast is an organelle surrounded by a membrane. And it has an internal structure consisting of many membranes. You may see these layers. And these contain the molecules, the proteins, the pigments that are involved in photosynthesis. And also they organize the interior space such that, for example, things can be enriched inside these membranes or outside. And these chloroplasts have their origin actually in cyanobacteria. These were the first who, and still existing organisms that perform photosynthesis in which oxygen is one of the products. Okay, and if we look at one of these membranes in a very schematic picture, we see a number of different proteins embedded in the membrane. This is the membrane, bilayer of lipid molecules, pretty well insulator. It doesn't conduct electric current. And the main players in this catalysis process are called photosystem 2 and photosystem 1 and cytochrome B6F complex. And what happens is the light excites a primary electron donor, as we call it, in PS2 and also in PS1, and drives an electron through the membrane. And this electron is then brought back through this B6F complex to a carrier protein to fill the hole that was created in this photosystem 1. And then the electron moves out there and essentially in the end enters a very energy-rich compound, NADPH, that is then used to in the synthesis of carbohydrates and other things. In all this process, the hydrogen ion concentration on this side of the membrane is systematically increased. And of course, here there is a more negative potential, here is a more positive potential. And the origin of all the replenishment of electron is happening in part of photosystem 2 by removing electrons from two water molecules, releasing O2, and of course adding the protons, the hydrogen ions to this space here. And of course, I will not talk much more about this because Michi is one of the world leading experts in how this happens. And you can certainly ask him if I have a question about that. Anyway, so this is essentially sort of a picture of the main players in this catalytic process. What it doesn't show is the large variety of light harvesting systems that exist in different plants. And these are basically antennas that increase the absorption cross-section for light of these chloroplasts, and they funnel the energy into photosystem 2 and photosystem 1. And in a more schematic picture, this is another thing I took from a textbook. You see the same players again, photosystem 2, photosystem 1, the proton pump, the cytochrome B6F complex, and the starting material is shown in green, water and CO2, and the products are O2 and carbohydrates, and the proton gradient. And this is a sort of symbolizing electron energy, and you have here electrons lifted to a high energy going through, filling holes in this thing, and here the electron is landing on this NADPH compound, and the energy difference, the binding energy difference for an electron between water and this is 1.2 electron volts, which is quite a lot, and is part of this rise in energy is contributed by light energy. No, the whole is contributed by light energy, but light energy is not 100% efficiently converted. But when people started to look at the details of photosynthetic systems, they didn't have, as I mentioned before, didn't have the tools that we have nowadays. And so that was middle of last century or even earlier. And so they were looking for organisms that may have, they also do photosynthesis, but may have simpler systems. And one of these is called photosynthetic purple bacteria. And it includes essentially an equivalent to photosystem 2, an equivalent to this proton pump, but no water splitting. Instead, they just recycle the electrons. So the light drives electrons in a circle, sorry, in a circle, and the result is the proton gradient that is coming out of this proton pump. And this, I guess, it has about 10% of the amount of different proteins than the chloroplast or the cyanobacteria, and therefore much easier to handle. And a lot of the basic observations were made with photosynthetic purple bacteria. And here's a picture of one of these called Blastochlorus viridis. I think it is in the order of about 2 microns in length here. And when it is grown under suitable light conditions, it develops a lot of infoldings of its inner membrane, and these are filled with photosynthetic molecules. And so it's a wonderful source for studying the function and structure of these. And so a lot of experiments were done, especially spectroscopy, laser spectroscopy, to figure out what happens in detail in this membrane and what do these molecules do. There was also, of course, biochemical analysis, what are the components, and essentially what was missing for quite a while was a three-dimensional structure of these, the molecules involved in photosynthesis. And especially the so-called reaction center, where the name comes from the observation that there is an antenna in which light doesn't cause any change. It just is handed to or is diffusing around in the antenna until it hits one particular molecule that is called the reaction center, where the reaction is happening. And so people were waiting for structure, and of course there were people who tried to develop methods to crystallize these, because crystallography was the only method at the time to determine the structure of such proteins. And but some people ran into some difficulties, and this is a response that one very, very prominent researcher in the field, George Fehr, got when he applied for a grant funding for finding conditions to crystallize the reaction center. And the idea of trying to crystallize the reaction center of membrane protein is ill-conceived and futile. The randomly intercalated detergents will make the formation of an ordered crystal impossible. The PI would be well advised instead to continue the beautiful work on the mechanism of crystallization of water-soluble protein. That was in 1979. Of course, George Fehr was not deterred. He found other sources of funding, but he had competition, and one of these was a man called Hartmut Michel. He worked at the Max Planck Institute, the same Max Planck Institute where I was. His boss, Dieter Oesterheld, supported the idea that he tried to crystallize membrane proteins, or they made a pact that Michel will try Oesterheld's favorite protein, but in return he can also try others. And one of the ones in which he had success was the plaster chloride pyridis reaction center. These crystals are in the order of half a millimeter or so in diameter, and maybe a millimeter or more in length, just roughly. But most importantly, they diffract X-rays very well. And that was a fantastic observation. This is a picture when you put the crystal in an X-ray beam and rotate it by a small amount, like half a degree or so. And it shows, first of all, to people who are used to looking at this, that the crystal unit cell dimensions are pretty big, and that the crystalline order is pretty good. The edge of this is in the order of 3 angstrom resolution. And Hartmut didn't have experience in the technicalities of determining structure from this kind of data. And so he was looking for someone, and lo and behold, he found me. So I was in the right place at the right time. And I also must have had already some kind of favorable reputation. Anyway, we got together, and we are still friends. I think that is a fantastic piece of luck, let's say, that we find a scientific collaborator that with whom you also get along personally. Anyway, we got to work on that together, and in about two and a half years, we came to a result, and that's shown here, the reaction center from Blastochlorus viridis. Here, this picture shows the proteins in cartoon representation, where the polypeptide chain is shown in a loop representation, except for where there is secondary structure like helices or beta sheets. And the reaction center consists of four different subunits, and they're called, for historical reasons, L, M, and H, and cytochrome. And this here shows a picture of an all-atom picture, an all but hydrogen atom picture, where one can, if we look at this for a while, you can see that this middle part has many more carbon atoms on its surface than the upper and lower part. And therefore, this indicates that this part has a hydrophobic surface, and that must be because it is situated in the membrane. If we turn this around, we get this view from the side. You see that it's relatively thin here, and so it has a elliptical cross-section. And the most important part, of course, are these, the so-called cofactors, or the functional parts. And these are four heme groups in the cytochrome. And then so-called bacteriochlorophylls in green, bacteriophytins, which are chlorophylls without a metal ion in the center, and then quinones, and an iron, and a carotenoid. And these are the carrier of the function. And I think the most surprising aspect of this structure for those people who were wondering what could be the structure that does this catalysis is the fact that it looks very symmetrical. So if you draw a line between these two chlorophylls and through the iron, you have basically the same arrangement of things, not 100% exact, but approximately the same arrangement on the left and the right. However, functionally, it is very asymmetrical. And as I said, people were studying this with time-resolved spectroscopy, and they found that once a photon has hit the reaction tender and brought the electron into motion, the time constants for these motion are shown here, 3 picoseconds from the origin to the first intermediate stage, then 0.7 picoseconds to the second, and about 200 picoseconds to the third, and then the missing electron here is replaced from the cytochrome up there So one of the hemes, the closest heme, is delivering an electron. So that there is no place for the electron to go back. And this is a sort of a general feature of these photosynthetic systems, that they use a lot of resources to prevent unwanted electron transfer events, because they cannot control where the electron will go if it wants to go back somehow here. So they immediately fill all the holes, and in the end, the electron ends up in this quinone. And this needs 2 electrons and 2 protons to be able to leave its binding site in the reaction center, and then be replaced by a new quinone, an unloaded quinone. And that's the way in which electrons get out of the reaction center. And so most people were very surprised that this part of the structure has basically no function. It's just there for apparently historical reasons. And so that was the biggest surprise of the whole thing. So this is a schematic again of what happens when light strikes this pair of chlorophylls with the different time constants. And here is shown the so-called cytochrome BC1 complex that strips the electrons and protons from the quinone and brings them back through the membrane. And the electrons are shuttled by a cytochrome to the bound cytochrome and then go back to it. So it's a circular, a cyclical pathway, and essentially the protons accumulate in this space up there. Alrighty, so that was essentially what we could discover and find out from the structure, together with, of course, data from spectroscopy and similar experiments. And towards the end of my talk, I would like to sort of describe what and not everything that happened since then, but a few things that happened since then. First of all, let us look at the reaction center core. That's this, what I call this group of cofactors, the chlorophylls, pheophytins, quinones. The iron, unfortunately, is not shown here. And this is part of these molecules. The chlorophylls and the iron are bound covalently to the rest of the protein. And here you see the amino acid side chains that do this. And then you see the subunits L and M, how they surround this group of things. And if you look from above, we see this, the pair of chlorophylls, not so very well resolved, the rest of the cofactors, but the transmembrane helices are arranged in the following way. If you go through the sequence, You first find this, second find this, third find this, fourth find this, and fifth find this. So 4 and 5 are interchanged. And the same is true for the M subunit. It's a general feature. And years later, structures of photosystem 2 came out, and here's a very incomplete list of papers. I want to talk about or use data from this paper here, Yumena and et al. From Michi's university, Okayama, and they were able to generate the best, as far as I know, best structure for quite a while for photosystem 2. And it's shown here, it's a monster. It has 20 protein subunits, 35 chlorophylls, and so on. But in the center, it has something that looks very, very similar to the bacterial photosynthetic reaction center. And but first, let me show you the cofactors. This group here is basically the same as the one that we find in the bacterial reaction center. This is a group of manganese, calcium, oxygen atoms that are the oxygen evolving complex. And again, I point to Michi to explain this if somebody has question. And here's a high resolution. This is one of the reasons why this paper became so famous, because it, for the first time, showed unequivocally that how this group of manganese and calcium and so on is constructed. And but let's look at the bigger picture, the central proteins called D1, D2 instead of L and M. And if we look from above, what we see is the arrangement of helices. If we look at where they appear in the amino acid sequences, again, 1, 2, 3, 5, 4. 1, 2, 3, 5, 4. So it's very, very similar to the bacterial reaction center. And here is a list of publications of photosystem 1 structure. And this, the first one already had a very high resolution, relatively high resolution structure of a photosystem 1. And it's also much bigger than the bacterial reaction center. Has 12 protein subunits, 127 cofactors. And if we look at it from above, we can see that it, we can assume that maybe there's in the middle is something that is similar to the bacterial reaction center, but it has a lot of peripheral things, and these are light harvesting parts that are integrated into the central 2 protein subunits. And if we look at the central cofactors, we see again they are arranged in 2 branches, but in this case they end up not by the quinones, but in iron-sulfur clusters. And so the electrons are actually going from the quinones to the iron-sulfur clusters. And in this case, the functional difference between the two sides is not as pronounced. There is some difference. So they are not identical, but maybe 30% go here, of electrons go here, and 70% here, but they all end up in the same place. And if we look at the surrounding helices, we again see 5 helices, somewhat differently shaped. And if we look at them from above, we see the pair of chlorophylls. We see an iron-sulfur cluster in the background. And in this case, the helices appear as numbered 7, 8, 9, 11, 10, because there are 6 transmembrane helices on each side, constituting the light harvesting proteins and being an integral part of these protein subunits. But again, there is this change in order, so it's 7, 8, 9, 11, 10, and the same here, which is a characteristic for all the structures I have shown so far. And in relatively recently, there was a bacterial reaction center crystallized and the structure determined that is similar to photosystem 1. In this case, it is perfectly symmetric. Electrons can go with same probability here or here, and they all end up with an iron-sulfur cluster. And if we look at them together, purple bacteria, photosystem 2, and heliobacteria, photosystem 1, we see the same principle, building principle, in all these 4 different types. Actually, these two are functionally very similar, and these two are also functionally very similar. But essentially, what this seems to tell, at least me, is that they all came from the same origin. It is hard to imagine that a convergent evolution, that means starting from different starting points, they would all end up in this way. It is more likely that there was an ancestor, most likely looking like this. And in the course of evolution, they diverged a little bit, but the main characteristics stayed the same. So that was basically my story. Again, this is the different way of representing the oxygen in the Earth's atmosphere. And again, here it is indicated, or these authors assume that about 1 billion years ago, the first plants or plant-like things appeared on the continents. Again, here is the great oxygenation event, which was many people who tried to imagine what that meant for the biosphere on the Earth. It meant probably a huge environmental catastrophe, because up to that point, there was no reason for the organisms at that time to worry about oxygen. And suddenly there's this contaminant, and but we are long, this is long behind us. And as I said, we would not be able to exist without this. And so in the end, I like to look at this picture, the sun, but it alone is not enough. There needs to be a catalysis system, and at the moment we are kind of putting the catalysis system in danger by systematically taking over our environment and making changes and contaminating it and so on. And if we want the sun to be on our side, we have to change our ways. Thank you very much.
Thank you very much for a great talk. So now it's open for discussion. Is there anyone who wants to ask questions? Okay, you.
Thank you very much, Professor. I'm Jerry from Tohoku University, Japan. Actually, I'm not in this field, but recently I've learned that some research on using artificial photosynthesis to for many purposes, such as producing hydrogen gas, fuel, and food. So my question is, what do you think are the possible ways that the structural biology of photosynthesis can contribute to the development of the sustainable energy solutions? Thank you.
Yeah, this is a very, very interesting question. I think I mentioned it during the talk that one of the pressures from evolution on the evolution of synthesis in the biosphere, one of the pressures is that it has to be the system has to be designed such that there is relatively little chance for radiation damage. So the plants and bacteria have to exist in very widely varying levels of intensity of light and so on, and they have to be prepared for this and that. And therefore, the efficiency, the sort of the cure for or the prevention of radiation damage was done at the cost of efficiency, and so the plant photosynthetic systems or bacterial are less efficient than we could imagine they are. So they use only a relatively small part of the energy. And the artificial systems can be, in principle, much better or better in terms of efficiency. And therefore, I think what you're describing has good chances.
Thank you very much.
Any other questions? You?
Okay, I have one question. I'm from Indonesia. My question is, usually in any biological system, to convert A to B, it doesn't only involve one specific enzyme, but many enzymes from one complex enzymes. And my question is what is the most easiest and most accurate way to determine the structure of these combinations of enzymes to form one complex system? Thank you very much.
Excuse me, I need help.
Can you repeat the question?
Okay. I mean, how to determine the structure of enzyme complexes that catalyze one reaction, one biochemical reaction. Because usually in biological system, to convert A to B, it doesn't only involve one enzyme, but like combination of enzyme that forms one complex.
You mean how complex enzyme complex contribute to the reaction, chemical reaction?
The structure, how to determine the structure of the enzyme complex.
How to determine the enzyme complex during the catalysis? How can we expect the enzyme complex? That is his question.
I'm not sure whether I understand what you're asking.
If a reaction requires a number of enzymes in a complex, how do you determine the structure of that complex?
Well, I mean, it depends on how stable these complexes are. I mean, there are transient complexes and then there are stable complexes. I think the current state of structure determination technology is much better in that, for example, cryo-electron microscopy allows you to look at a whole sample of different states of a protein or a group of protein. And I could imagine that if there are complexes formed in the course of a series of reaction, and you do it the right way, you could essentially catch different complexes in essentially one sample simultaneously.
Okay, thank you very much.
Questions? Hey, you? And you'll be next.
Hello, Professor. I'm Hao Liang from Gifu University. So, as you mentioned just now, recently the cryo-electron microscope is widely used to detect the protein structure. But so, do you think it will completely replace the X-ray crystallography? So, or currently, X-ray crystallography still has some more advantage point that cryo-electron microscope cannot replace it. Okay.
Yeah, that is a good question. At the moment, a limitation of cryo-electron microscopy is that it requires samples of a certain size. So if your molecule is too small, it will not create enough contrast in the electron beam, and you kind of cannot study the structure. So you need it's kind of almost a little bit against. It seems like against logic, but you need a certain size to be able to study the structure, and so that, of course, is a limitation that can be filled by crystallography. Otherwise, I think cryo-EM has so many advantages that people will always try for suitable objects. Will always try to do this first, and perhaps there are new ways of crystallography. And again, I point to Michi because he's doing things that are very different from what we did, and meaning that the requirements for crystals are very different than what he is interested in really tiny ones, less than a micron, and we were interested in big ones, about a millimeter or so, but it enables him to do, for example, time-resolved crystallography, which is not possible, of course, with cryo-EM.
Okay, thank you.
That was a very nice talk, thank you. I always had terrible trouble understanding photosynthesis when I was an undergraduate. I mean, it was deeply mysterious then because we didn't have these beautiful structures. But I was just wondering, when you were talking and giving these timelines, where did this come from? Is this a very ancient thing? When do we know, have any idea when chlorophyll was first invented? I mean, have bacteria always been using sunlight to harvest energy, or it seems to me they used to be in the deep sea vents and they sort of just used chemistry rather than photons.
That is I think, an open question. Except that the appearance of oxygen in the atmosphere, as I said, is very hard to imagine without a suitable catalyst for using sunlight. And so far, there's no other process known or even considered that does this except for photosynthesis. So, but essentially, I mean, in order to become use oxygen or use water splitting to replace electrons, you already have to have reached a certain pretty high level of evolution. And so that means that people assume that long before the oxygenation event, there was already a chlorophyll-based type of photosynthesis. And
use some other electron donor than water, I suppose.
So they, as I said, everything lived in water at the time. And for example, these purple bacteria, they don't evolve oxygen. They even, I mean, the one we use is actually, for it, oxygen is a poison. And so it can totally live with, or it wants to live without sight of oxygen. And I guess similar systems may have existed before the oxygen or the water splitting was invented, let's say. Yeah.
Any other questions from the floor? If not, I will ask one small question. So you showed the structure of reaction center and the photosystem too are very similar, even during the evolution of 3 billion years.
Yeah.
In the universe, if there is a planet other than Earth that utilizes the sunlight, and if there is a photosynthetic organism exists, do you think that organism share the same structure with the reaction center you analyze?
I missed the crucial part of the question.
Yeah. So if there is other planet that has biological organism, photosynthetic organism, and completely different planet, do you think the same structure exists in that biological organism?
I think this is arbitrarily unlikely. because I think that the reason why these are all looking very similar is that one successful organism took over the whole biosphere, and but it could have started in a different place, and then that would have taken over. So I don't think there is anything really so unique that one couldn't imagine other ways of doing photosynthesis. Yeah.
Ready. Thank you. Okay, so now time is approaching. So let's thank him again.
Thank you very much, Dr. Deisenhofer and Dr. Suga. Let's show appreciation with another round of applause.